Hello and welcome back as we begin our look at the digestive system, which will end up being a four-part series of really trying to understand structure and function within the digestive system. And as we look at structure and function, we will also spend some time reviewing the histology, right, of the digestive system. So we're not just looking at being able to identify organs when we talk about the structure, but we're also looking at and talking about being able to relate the function to the histology, which we know the histology is going to drive the structure of the digestive system as well. And so I would kind of like to start our look at this by considering something within the digestive system. That was not the right color. So before we do too much with the digestive system, I need you to understand and appreciate that our digestive system is developed and composed much like a donut. And what I mean by that is we have a circle, and then in the middle of that circle, we have another circle. In other words, we have a tube within a tube, or we have ourselves a donut. Right? We have ourselves a donut. And within this region of our donut, within the doughy part of our donut, we find the organ systems. Right? So we're finding the cardiovascular system, we're finding the nervous system, we're finding the skeletal system, the urinary system. We're finding all of these various systems. We're even finding various organs. We're finding the liver, we're finding the spleen, we're finding the pancreas. All of these things are within the doughy part of our donuts. Then we've got this middle section that is located right here. Right? We have the donut hole, if you would. And it's within the donut hole where we have the gastrointestinal tract. Right? And so that hole, the donut hole of our donut, is what is really made up of the esophagus, the stomach, the small intestines, the large intestines, and the rectum. That is the inner tube to our donut. That is the inner tube to the larger tube that is surrounding it. And so kind of understand, kind of get this visual, and as you have this visual going of this tube within a tube, where you've got this outer ring that has all of the structure and organization and day-to-day function, and then passing through it, you've got this hole that allows things to pass directly through without ever coming in contact with the stuff that is in here. Right, so in other words, if something is in here, right, it has to be brought in. It has to be absorbed. And if it's not absorbed, it is simply going to pass through. It's going to come out the other end just as easy as it came in through that way. And so it will never come into this larger tube. So what does that look like for the whole of the body? Well, let's kind of take what we just looked at and apply it here. So here's the external environment. Here's the oral cavity. We come down into the esophagus, down into the stomach, down into the small intestines, down into the large intestines, down into the rectum, and then we exit through the anal canal. And so this outside of the body is, if we go in reverse order, directly linked to this. And it's linked with this tube. And that tube is designed to separate out what is allowed to come into the body, what is allowed to pass through the cardiovascular system, the lymphatic system, over into the liver, that's passing through the kidneys, that's going into the nervous system, that's feeding and supplying our skeletal muscle. All of that is being regulated by what can and cannot be absorbed through this segment of the gastrointestinal tract. And in fact, that is one of the functions of the digestive system. Within this gastrointestinal tract, we are going to move nutrients, water, and electrolytes from outside of the body inside, from external to internal. And we're going to do that through both chemical and mechanical digestion. And so if we look here, we see this term autodigestion. We are going to be doing autodigestion through the use of enzymes. In other words, the peanut butter and jelly sandwich that you ate for lunch, right, does not enter into the small intestines looking like a peanut butter and jelly sandwich. It has been digested, and it's been digested along the way. Now, this tube within a tube that we define as being the gastrointestinal tract actually has segmentation to it. So along the way we have regulatory gates. Right? In other words, the esophageal sphincter, right, regulates what goes from the esophagus to the stomach. The pyloric sphincter regulates what comes from the stomach into the small intestines. The ileocecal valve regulates what comes from the small intestines to the large intestines. The internal and external anal sphincters regulates what goes from the rectum and passes through into the anus to be gotten rid of through the body. And so we have all of these little checkpoints before things pass to the next area. And we're going to kind of play on that a little bit more because, as we will find out, what is happening here regulates what happens within the small intestines. What's happening here is going to regulate what is happening down here in the large intestines. And we'll touch on that more. The other major function that is happening here is mass balance. Or another way of describing mass balance is fluid balance. And so sometimes you'll see this referred to as fluid balance. And on the average day, you are either consuming or producing approximately 9.3 liters of fluid. Right? So how are we producing or consuming 9.3 liters of fluid a day. And why do we have to keep a balance of this? Well, if you look right here, right, this is the amount of fluid that you are consuming on average on a daily basis. Now, by consuming, we mean whether you're drinking a bottle of water, whether you're having a cup of coffee, whether you are enjoying a rum and coke while you are at home. You are consuming that. Right? The hamburger that you had, the peanut butter and jelly that you had, the bowl of cereal that you had, all of that stuff has fluid that's within it as well. And that contributes to the amount of fluid that you are taking in. But that's not the only means by which we introduce fluid into the digestive tract, because indeed our body, along the gastrointestinal tract, we are also releasing other fluid. So your saliva is water-based, and you're producing about 1 liter of saliva a day. That's entering into the digestive tract. Your stomach is producing about 2 liters of fluid a day. That's entering into the gastrointestinal tract. Your liver is producing bile at about a rate of 1 liter per day. That's getting introduced into the gastrointestinal tract. Your pancreas is producing about 2 liters of fluid a day. Your intestines, mainly through the secretion of mucus, is producing about 1 liter of fluid a day. And so you've got about 9.3 liters, or the equivalent of 4.5 2-liter soda bottles a day. Right? And of those 9.3 liters of fluid being produced today, or a day, 7 of those liters is coming directly from you. 7 liters a day your body is producing. And so imagine if there was not a mechanism in place to recoup that fluid. You would dehydrate. Your cells would stop functioning appropriately. And so that would not be, that's not beneficial. Right? That is not good. And so we have to have a way of, if we're getting rid of 7 liters of fluid a day from within us, but we're only ingesting 2.3 liters, your body has to be able to conserve how much fluid it has within it. And so it's got to work to reabsorb that. And so that is where the small intestines and the large intestines comes back into play, because you can see that the small intestines is working to absorb about 8.3 liters of that fluid. The large intestines, to a lesser degree, it's absorbing about 0.9 liters a day. And so we conserve over 95% of the fluid. We are conserving, we are reabsorbing. And our feces, the end product of digestion, is we're really only losing about 0.1 liter. Now, this is not taking into consideration what the urinary system is doing, because naturally your urinary system is saying, well, all right, we're ingesting on average 2.3 liters, but what if we ingest 5 liters a day? We have to be able to compensate for that. And so that is where your urinary system comes into play. And by the way, the other thing that I did forget to mention here was why the importance for having this tube within a tube. And the answer to that is to prevent things like bacteria, pathogens such as viruses, allergens from entering into the larger tube. In other words, into your body. And so if that bacteria cannot be absorbed, it doesn't get into the system. Not to mention, as we will find out, down in this area here where we are dealing with the large intestines, it has its own natural biota of bacteria. And so we don't want to be disrupting anything along those lines either. And so we really try to exclude through this process what is harmful and only extract and conserve what is needed. And the way that this all takes place is through a number of mechanisms. Right? So through digestion, right? Both chemical and mechanical digestion, through absorption, which happens in the small intestines, through the movement of this material, right? That helps to absorb, especially fluid balance or maintain fluid balance, and then secretion, getting rid of whatever is extra. Right? And so the processes that we're really dealing with is that we have to consider as we go through the digestive system is digestion, absorption, motility, and secretion. This slide here is pretty much just a overview of the organs of the digestive system. All right. I am not going to spend much time on this. This is really a review for you of what we've already looked at in lab. I will say that keep in mind just a couple different things. The esophagus. Right? The esophagus is made up of both skeletal muscle and smooth muscle. Skeletal muscle is found closer to the oral cavity, all right, whereas smooth muscle is really located within the lower 2/3 of the esophagus. So skeletal muscle, the upper 1/3, smooth muscle, the lower 2/3 of the esophagus. The other thing that you want to keep in mind is that the small intestines are really not working by themselves. So they are working in conjunction with the liver, the gallbladder, and the pancreas. And so the reason why those three structures here are in red is because they are considered to be accessory structures, just as the salivary glands are accessory structures and the teeth and the tongue are accessory structures. So all of those are accessory structures to the digestive system, which is why they are in, once again, they are highlighted in red. And the other thing that we'll get into that in a few minutes. Hold that thought. And so be aware of this, be able to identify things. You never know what I may or may not put on the exam. So again, the gastrointestinal tract specifically is made up of the esophagus, the stomach, the small intestines, the large intestines, and the rectum. That is the tube within the tube. It does not include the oral cavity or the anus or the anal cavity. And again, this here is just highlighting those accessory structures as well. So we've kind of already touched on that. We don't have to spend too much time on that. Just be aware that tongue, teeth, salivary glands, liver, gallbladder, and pancreas are all accessory structures, and we will be spending a lot more time on the liver, the gallbladder, and the pancreas in future videos. Saliva. I want to talk a little bit about what saliva is. There's two types of saliva that we have. We have saliva that is produced that directly directly is involved in digestion, right? In other words, chemically breaking down food. And then we also have saliva that is simply there to lubricate and moisten. Um, and we'll talk about what the difference of that is here in a couple minutes. Not too much longer, but a little bit. So saliva that is involved in digestion contains enzymes. Right? Enzymes such as salivary amylase. Right? Salivary amylase works within the oral cavity and the esophagus to break down starch. Once salivary amylase reaches the stomach, it gets inactivated. It gets shut down. In other words, the pH change inhibits the work of salivary amylase. But another enzyme that is released from within the oral cavity is lingual lipase. Now, lingual lipase is inactive in the oral cavity, but it does become active once it reaches the stomach. Right? And part of what activates it in the stomach is the acidity or the pH. And so it is activated by a change in the pH. So it's released in the oral cavity just as salivary amylase is, but lingual lipase does not function in the oral cavity. It's only functioning within the stomach. Right? Salivary amylase is functioning in the oral cavity, but it is inhibited in the stomach. So a lot of this is driven by pH. Right? By the way, lingual lipase does not technically digest fats. It goes through a process of what we call emulsification. And emulsification is where we're taking these larger fat globules and we break them down into smaller fat globules. Now, within saliva, we also have water, but we also have some other components to saliva in addition to just having enzymes. So mucus, right? Mucus is there to help lubricate the food. It is there to help move the food through the esophagus so that it doesn't damage the epithelium. In other words, some food we eat is abrasive. And if we didn't have mucus that was helping to lubricate it, it could cause damage to the stratified squamous lining, the non-keratinized stratified squamous that lines the esophagus. Notice this term bolus. Right? So food goes in as food, but once we begin to chemically alter that food, it's no longer food. So the peanut butter and jelly sandwich that you had for lunch goes in as peanut butter and jelly sandwich. But once it hits the esophagus, it's no longer peanut butter and jelly because it's already started to be broken down. And so we define that as being a bolus. Right? So bolus is what that food is as it's traveling down the esophagus, getting ready to go into the stomach. Some other things that are in saliva, we have lysozymes, which are designed to kill bacteria. We have immunoglobulin A, which inhibits bacterial growth, what we refer to as being bacteriostatic. Right? In other words, it inhibits bacterial growth, but it's not completely killing it. Lysozymes, on the other hand, are bactericidal. Right? Bactericidal does kill bacteria. Right? Does kill the bacteria. So bactericidal kills the bacteria. Bacteriostatic inhibits the growth. We've got both of those aspects built into our saliva. And then you can see we have electrolytes, and that basically helps to maintain composition of the saliva. In other words, helps with pH balance and other such things. Now, remember I said there was two types of saliva. And those two types of saliva are produced by two different types of glands. And so one type of gland is what we define as being intrinsic glands. These are glands that line the oral cavity and helps to keep the mouth lubricated so that you don't dry out. Right? And the other type is extrinsic glands, and it's these extrinsic glands where we actually find the digestive enzymes are released. Right? And so these guys are actually producing the enzymes. Right? So things like salivary amylase, and lingual lipase, right, are being secreted through these extrinsic glands, as well as mucus and water. Your immunoglobulin and your lysozymes are more so being produced and secreted as part of the intrinsic glands. Right? Now, your extrinsic glands, of course, right here, you've got your parotid gland. Right here, you have your submandibular gland, and then located right up into this area here, you've got your sublingual gland. And they are again your three main salivary glands that are involved in the production and the release of salivary amylase and lingual lipase. And so we're going to stop here for right now. And give you time to digest this information and kind of go back and rewatch anything that you need to rewatch. And we'll see you back here in a few minutes for part 2 of the digestive system.